Good morning. We are going to be doing lecture 3 today. So lecture 3 is called cells. So let's begin. In the first lecture, we talked about the characteristics of living things, right? And one of those characteristics is organization. Okay? There are some complexity to life, and the organization is going to hold true all the way to the molecular level. So we learned in lecture 2 that everything in the universe is made up of elements, and the smallest form of an element is atoms, right? So we know there are essential elements that makes up life, right? 5 of them. And these things can come together to form small molecules, like glucose, like water, right? All these things are quite small. They have, just a handful of atoms in them. For water molecule, for example, there are only 3 atoms. For glucose is a little bit more, 24, but nevertheless, still quite small in numbers. And then later on in lecture 2, we talk about the macromolecules, right? These are the carbohydrates, the proteins, lipids, as well as nucleic acid. And these are polymers.
Morning, sir.
Good morning. These are polymers. They're very, very long. They have hundreds and sometimes thousands of atoms in them. Okay, and so today we are moving up this ladder, right? We are going one level higher to see the next level of organization. Okay, and that is organelles and cell. And in fact, we're going to spend quite a bit of time talking about the cell for a while in the course. Okay, all the way up to like lecture 9, we'll be focusing on various things that are happening within the cell. But today, this particular lesson, we will learn the features of a cell first. Okay, what makes up a cell? What is a cell? And what are some of the structures which we call organelles that are found inside the cell? Okay, so organelle, you can think of them as mini organs. That's literally what it means. They were tiny little organs that are found inside the cell, and they carry out specific function for the cell. So that's what we are going to be focusing on today. Now, I know you do have a math quiz as well as a bio quiz today. So I will try to cut the lesson short so that you have some time to prepare. So we'll probably skip the break and then just push through, and then I'll let you go a little bit early. Okay, hopefully that will be helpful. Okay. So this picture shows the relative size of things. All right. So we as a grown adult, height is usually about 1.5, 1.6. People are like 5 feet, 6 feet tall, right? And that's what a meter is essentially. And then you can shrink down the size by dividing by 10. So if you take a meter and you divide by 10, then you have like 10 centimeter, right? Which is roughly the size of like an ostrich egg that's really big, and then chicken egg is a little bit smaller. I'd say it's about like 5 centimeter-ish, right? And then you can keep on dividing by 10 to go smaller and smaller. Eventually, when you get to the micrometer range, that is where you will find the cells. Okay, so animal cells, plant cells, they are between 10 micrometers to 100 micrometers. And just to remind you what micrometer is, 1 meter has 10 to the 6 micrometer. That is 1 million micrometer. So it's quite small, right? It's 1 million times smaller than a meter, right? This is a meter. If you divide it by 1 million, then you get a micrometer. Still smaller would be organelles and bacteria. These are structures that are, like I said, found inside the cell, and so they, instead of 10 micrometers, they're just like 1 micrometers. And some bacteria, they could go inside your cell actually and cause problems. Some can stay out of the cell, but in terms of size, they are similar to that of an organelle. Okay. If you go to the nanometer range, okay, 1 meter has 10 to the 9 nanometers. So now we are talking about a billionth times the size of a meter, right? Okay, and that's where you will see viruses, proteins, and lipids, the macromolecules essentially. So to visualize things that are in the a micrometer range, we can use a light microscope. All right, so these microscopes are very common. You actually will do a virtual lab to see how to use it. But like if you were in person, right, like you would be able to handle those microscopes, right? Those microscopes are fairly cheap, like about, $200 each, the ones that we have in the lab. But then if you want to see like really small things, the virus and stuff like that, then you need an electron microscope. And these are very expensive, okay? Some of them are as expensive as like a luxury car, right? So what is a cell anyway? You hear people talk about cells all the time, right? So cell is defined as the smallest unit that has the properties of life. You, of course, know about the property of life, right? Like it's going to be on your quiz today, right? So, there is the ability to maintain homeostasis, right? Energy requirement, response to stimulus, all these characteristics will still be there on the cell level, okay? If you go inside the cell, right, let's say we pick a macromolecule, right, like protein. Protein is no longer considered as alive because it doesn't have the properties of life, right? It's not going to reproduce, it doesn't require energy, right? So the smallest unit that's still satisfied, that checklist, is a cell. You cannot go smaller than a cell and still expect to have those kind of properties. Does that make sense? Now here we have 3 pictures that shows you different kinds of cells. Does anybody want to guess what this what this first picture shows? What kind of cell do you think this is? This usually people know. That one is quite obvious. You've probably seen it in movies and whatnot. Kiana said brain. That's right. That is the brain. These are neurons, right? And they're making connection with each other. Over here, these blue balls, those are streptococci, which can cause things like strep throat, right? Okay, and then here, this one is an amoeba. Amoeba, okay, which is like a pond-living organism. And sometimes they find their way into swimming pools. And if it gets to kids or even adults sometimes, then you get diarrhea and sick from it. The point from these pictures is that different cells look very different. Okay, they do look like jellyfish, don't they? But these are really small. These are microscopic. Actually, you cannot see them from your with your naked eye. But they look very different. Okay, but there are certain features that are common for all cells. Okay, even though the brain cells look nothing like these streptococci, and they look nothing like the amoeba, there are 3 characteristics that are shared by all kinds of cells. So the first one is that they all have a cell membrane. A cell membrane is also known as a plasma membrane, and that is a barrier that separates the outside environment from the inside of the cell. Okay, as we will see later on, it's important to have a barrier that regulates what goes in and leaves the cell. Okay, you can't just let things randomly come in and go out as they please, right? You will lose valuable nutrients that way, or you might be bringing in harmful things, right? So by having a barrier, by having a cell membrane, you help regulate that. Okay, so that's the first common feature. I'm going to ask you this later, okay? So make a mental note. First common feature, cell membrane. Number 2, all cells are going to have some kind of genetic material. Okay, so we are familiar with DNA and RNA, right? So those have hereditary properties. Okay, when a cell divides, it passes on those genetic material to the next generation. Okay, and finally, inside the cell, it has this gel-like substance. Okay, it's kind of I would say it's kind of like hair gel, the consistency. It's not really like water. Water is like too liquidy. It's kind of gel-ish looking. And we call that the cytoplasm. Cytoplasm, right? So the cytoplasm is where everything else is floating in there. All right. So here we have a cell membrane, we have the cytoplasm, and these things here that are floating around, those are the organelles, right? Organelles. Okay? And sometimes they might have a nucleus. Okay, they might have a nucleus in the middle to hold together the genetic material. Okay, but a nucleus is not a common feature of all cells. Some cells don't have it. Okay, it just happens that this picture has a cell that has a nucleus. Do you understand? Okay, so cell membrane, genetic material, and cytoplasm. Those are the three key features of all cells. There is something called the cell theory, and there are 4 parts to the cell theory. Okay, they describe different aspects of cells, basically. So the first part of the cell theory goes like this. Every living organism consists of 1 or more cells. Okay, 1 or more cells. There are some organisms that are just 1 cell big. For example, the amoeba we saw earlier, that's just 1 single cell. There is something called paramecium. You'll hear me talk about paramecium several times in the lecture. That's another single cellular organism, okay? Just all by itself, 1 whole cell. They have little hairs here that help them swim around. Okay, they have a little central area to keep the DNA, right? So that's just 1 cell. Some organisms are bigger, right? This is still invisible to the naked eye, but it has 3,000 cells. This is C. elegans. It's a kind of microscopic worm that scientists use to do experiment on. Okay, scientists have these things called model organisms that they know a lot about. They know all their genetic makeup. They know all the proteins that they make, right? And they know a lot about them. So that makes them ideal to do testing on them, right? Test for new drugs, test for a new therapy, so on and so forth, right? So this C. elegans is an example. So one of the things that I've read recently about experimenting with C. elegans is that they found a gene. Okay, a gene is a segment of DNA. Okay, we learned that later, but for now, like it's a segment of DNA that controls some aspect of the organism. Okay, so they found this gene, which if they mutate, if they change it, then the lifespan of these worm almost double. Okay, so that's very exciting, right? For I don't know, for various reasons, I guess for people who are trying to find ways to extend lifespan. Okay, so they found out, oh, humans also have this gene, right? Maybe that has something to do with extending lifespan. Okay, so what they could do next is find people who live very old, right? We call them centurions, right? People who live over 100, and then maybe look at the gene in those people, right? See if there's any kind of correlation, right? So a lot of study start with these model organisms, okay? So only 3,000 cells-ish, okay? And when it comes to humans, okay, we are like super big, right? We have a lot of cells, roughly 37.2 trillion cells, plus or minus, depending on how big you are, right? Okay, so the point is, it doesn't matter. You can have 1 cell, which we say unicellular, or you can have many, many cells, which we call multicellular. All right, so that's the first part of the cell theory. If it's alive, it's gonna have cells, at least one cell. Second part of the cell theory. A cell is the smallest unit of life. We know that already. I explained that to you earlier. Individually alive, even as part of a multicellular organism. Did you know you can take cells from your body and you can put it in a Petri dish. That's what these are. These are little circular container. Those are called Petri dish. And you can supply nutrients to them, right? That's what the red solution is, right? It has like glucose in it, fructose in it, it maintains the pH and so on and so forth. So you can supply them with nutrients and the cell will grow in the dish without you, okay? So individually, on their own, they can grow without you. Not really like cloning. Cloning would be like, now this is not true, okay? But like cloning would be like taking a cell from you and then growing the entire you. You know what I mean? Like then you would end up having like someone that's exactly like you, right? So that's not what this is about. This is just talking about taking cells from your body, and then they can be like your pets, okay? They can be alive on their own in the dish. Does that make sense? Now, they don't live forever though, right? Okay. Like if I take some cells from my, I don't know, from my cheek or something, and then I grow them in the dish, right? They're gonna, even if I keep on giving them the best nutrients, okay, they will only grow for like maybe a week tops. Okay. So most cells can live on their own outside of the body, but they won't live forever. Okay, they live longer in the body most of the time. Can it grow a new part of you? Okay, that's a good question. On its own, no, okay? But then like if you do some things to it, and that's like beyond the scope of this lecture, okay? But like sometimes it's possible to grow like you know how people have accident and then they lost a ear or something, right? It's possible to grow the ear back and then attach it back on using cells, okay? But not like a functional one, okay? But like for cosmetic, right? Like it's gonna be made of like tissue, right? So you can look that up, right? If you're interested. All right. So I was telling you, right? If you take the cell and then you grow it in the dish, it's not gonna live forever, right? It's gonna die eventually. But sometimes cells becomes immortalized. Immortalized means it won't die. And that's the case with cancer cells, right? Okay. So there is this thing called a HeLa cell. HeLa cell is a cell line. All right. There are many different cell lines out there. Let me just explain to you what they are, in the broad sense. Cell lines are basically immortalized cells. They grow forever. As long as you give them nutrients, they will keep on growing and growing. If they take up too much space from a dish, you can take some off and then put it in a new dish, and then it will grow and fill up the whole dish. Okay, so biotech companies, they have a lot of different cell lines. Some are taken from liver, some are taken from lung, and so on and so forth. Okay, skin cells, right? And they sell them. Okay, and you can use them for many things. Okay, you can actually use them to study medicine, right? If there's like a new drug, right, you're not going to test it on humans directly. You're not even going to test it on animals directly. The first step you have to do in testing a new drug is to test it on the cells, right? Okay, because cells are easy to grow, easy to control, easy to maintain, right? So you can do your experiment very quickly there. Okay, but these cells, you can use them to make vaccines, actually. Okay, so a lot of vaccines, they use a cell line that has cells called like fibroblast. Okay, and they're used to study many things, like cures for cancer in some cases as well. So that's what cell lines are. Okay, like you can buy them if you want, but I mean, like you wouldn't have the means to maintain them, to grow them. Okay, unless you're in a lab. But anybody can buy them. So one famous cell line is called the HeLa cells. Right? HeLa cells, right? And it's called HeLa because the cells was taken from this lady. Okay, her name was Henrietta Lacks. That's why it's HeLa cells, right? And this lady, she had ovarian cancer. And at the time, the doctor took a little biopsy, a little sample of her tumor, and without her consent, he tried to grow a little bit of it in the dish, just out of curiosity, just to see what happens, right? Okay, and to their surprise, it just keeps on growing; it won't die, right? Unlike the cheek cells I was talking to you about, right? Like normal cells, we put in a dish, it grows for maybe a week and then it dies, right? But these things just keeps on going and growing and growing and growing. Okay. Long story short, eventually, they commercialize this HeLa cell and then they sell it right? For research purposes, right? So she died of her condition later on, but her cells lives on, right? Her family was not happy about this, right? They sued the company. The company that selling this is Thermo Fisher, and they're not happy with the company. They're like, you are profiting off our mom, I think maybe, right? Or aunt, I don't quite remember the whole story, right? Without her consent, right? So, like this is not right. Right? And the company tried to dismiss their lawsuit. And I think the end result is that the judge rules in favor of the family. Okay, so they got some compensation as a result of this. Ramona asks, so cancer cells are immortal? Yes, essentially that's what they are. They are immortalized cells that keeps on growing and growing, and they takes up a lot of resources in the body, right? And that's why, it could kill the organism. Okay. We'll learn more about cancer later on in the course. Do you guys with me? Any questions? All right. So the 4 parts of the cell theory, right? Number one, um, all living things are made of one or more cells. Number two, individually the cells can be alive on their own, right? Durrett asked a good question. Is this same as stem cells? So stem cell is a type of cells. If you take stem cells out of the body, you can you can grow them in a dish, and you can use different we call them growth factors, okay, or stimulants to help them become a particular kind of cell. Okay, so for those who don't know, stem cells are cells that can become any cells of the body. They can become heart cells, lung cells, brain cells, so on and so forth, right? And as you might or might not know, brain cells don't come back once they die, right? So if we know how to change the stem cell, what condition we have to give to the stem cell to cause them to become a brain cell, that's very useful, because then now we can reintroduce the brain cells in the brain to repair the damage, right? Okay. Yep. Stem cell treatment is very common for things like leukemia. Okay. Where they transplant the bone marrow, which contains the stem cells, right? And there are a few other experimental stem cell treatment. Now, any mothers here? If you are a mother, when you gave birth to your child, in Canada, there is an option to freeze the cord blood, right? From the placenta, from the umbilical cord, right? Do you guys know what I'm talking about? Anybody? Yeah. So like the baby connects to the mother through a cord, right? An umbilical cord, the one that you have to cut when the baby is born, right? And there is a little bit of stem cell in there of the baby. So you can choose to store that, freeze it, right? And in the future, they're saying if your child becomes sick of some disease, maybe the stem cells can be used as a therapy to treat them. Because the stem cells is from their body, it would only work for your baby, right? It would only work for your child, right? So yeah, it's crazy expensive, I think. You have to pay an upfront fee, and then you have to pay annual fee to keep them stored, right? I didn't do it. Like it was just like too expensive. Like it's like I don't know, the benefit, the potential benefit doesn't really justify the immediate cost for me anyways. But some people do that. Some people like to, you know, do everything for the kids, right? Anyway, I went on a tangent. Sorry, I always do that. Let's go back to the lesson here. Number 3, part 3 of the cell theory is that all living things come from the division of pre-existing cell. Nobody knows where the first cell came from. I mean, there are theories about it, but nobody knows for sure, right? But we know now that in order to get new cells, right? In order to get new cells, the cell must split. Okay, so in humans, last time we said we have 46 chromosomes, and when they split, they copy all the genetic material, and in the end, you will get 2 identical cells. And so this here is called the parent cell, and then these are daughter cells. daughter cells. Okay, so all living cells, come from division of pre-existing cells, and cells containing hereditary materials, the DNA, is going to be passed on during the division. Okay, so this process is called mitosis, and we will learn a lot about it later on in the course. I keep on saying that, right? It's like I keep on telling you, oh, we'll learn this later, but we will. It's coming. Anybody know what we're looking at here in the picture? What? It's a kind of plant. What is it? Oops. What is it? Anybody? Onion, exactly. This is onion, right? So if you take a piece of onion cell and then you stain them, you will be able to see all these activity. So here you can see a cell. Oh my goodness. You can see a cell that is dividing, right? It's splitting into two, right? So that illustrates the point number 3 here, right? All cells come from division of existing cells. Okay. We cannot see cells because they're really small, right? In the micrometer range, right? I was telling you about it. And so to study it, we have to use a microscope, right? We have to use tools to extend our senses so that we can make observations, right? So the first guy who sort of invented the very primitive microscope, I guess, is this person right here, okay? Leeuwenhoek, that's how you pronounce his name. Okay, so he had a handheld microscope that looks like this, right, with a few lenses, and he was looking at pond water. He put a few drops of pond water, and then he was looking through it at it through this device, and he noticed there is a world that exists beyond our senses. Like nobody knew that there's so much life in pond water, right? Like you look at the water in the pond, like, okay, like I don't see anything living, but deep down when you zoom in, is like a whole community of living organisms, right? So that started the whole field of microscopy. Okay, and nowadays, right, this is a typical light microscope. Okay, and we can use it to see things that are in the micrometer range. If you want more details, then you need to get an electron microscope, right? These are, like, once again, very expensive stuff. Okay, so if you want to go down to the smaller end on the micrometers, or sometimes even in the nanometer range, then you have to use electron microscope. So on the left side, we have a transmitting electron microscope, which is good for looking at cross section of stuff. Like cross section is like if you have a specimen and you cut it, and then you look at the cutting, right? Okay, and then the scanning electron microscope is to produce a 3-dimensional view of the specimen that you're looking at, right? So this is very good at studying structure, right? On the surface, while this is very good at looking at the structure on the inside. Does the college have these? No, these are very expensive, right? Like I said, it costs more than a sport car. We don't have that kind of budget, right? But hospitals, very common. Universities, very common. Anybody want to guess what we're looking at here? Something in the body. You guys don't like these guessing games, do you? It's okay, there's no wrong answer. Just use your imagination, right? What is it, sorry? Someone said something.
I think it's the cells in the intestines, I think.
Cells in the intestine, good guess, right? We have stem cells, very good guess. Lung, villi, okay, yeah. Yeah, I mean, like, these are legitimate guesses, right? I totally can see the rationale behind what you're saying, right? Okay. But the correct answer is they are cilia. Cilia in airway. Okay, cilia and airway. Cilia are these little hairs, okay, that moves around. We breathe in a lot of like pollutants and allergens, right, from the air, and we want to filter them, right? Okay, so the cilia is going to move like this and going to sweep up all the dust particle, and then you have like a phlegm, right? That goes up to your throat. As you spit it out, you swallow it, right? That's what they're for. People who smoke a lot, heavy smokers, they damage their cilia. They have no way to remove all those trapped particles. And so they have smoker's cough, right? You can hear smokers, having these very heavy coughing, right? And then and they spit out the stuff afterwards, right? That's because they don't have the cilia. Cilia are also found in the fallopian tube of the female body, right? Fallopian tube, of course, connects the ovary to the uterus, allowing the egg to go towards the uterus. And the cilia in the fallopian tube will help guide the egg, right? It's like, come on, move this way, right? Otherwise, they don't know which way to go, right? So that's what the cilia is for. And that's what they look like, okay? All right. I have been talking for half an hour already. Let's see if you learn anything from me. Which of the following is a concept of the cell theory? Okay, why don't you read it? And then I will launch the poll for you. Anyone else? All right, I'm just gonna end it right there. Yeah, most of you got it right? It's D. A cell is a basic structural and functional unit of living things. Very good. Okay, moving on. This one. Okay. The correct answer is the nucleus, right? Let's go back here. Right? Common features of all cells: cell membrane, genetic material, and cytoplasm. Common features of all cells: cytoplasm, genetic material, and plasma membrane, right? So the correct answer is C. Okay. Not all cells are going to have a nucleus. All right. So we are now going to go into the cell and we are going to look at different structures associated with the cells. But before that, we should know that there are two categories of cells. Okay, we have cells that are more complicated, and then we have cells that are like simpler. So cells, we have two kinds. Okay, eukaryotic cells and then prokaryotic, okay, cells. Sometimes they are called eukaryotes, Y-O-T-E, and prokaryotes. We are made up of eukaryotic cells. They are very complex. They are generally bigger. And most importantly, we have a nucleus. Okay, so the nucleus is the central area that contains DNA. Okay. inside our cells, inside eukaryotic cells, there are many different kinds of organelles. We have mitochondria, ribosomes, Golgi body, so on and so forth. A lot of these organelles for specialized function. We will learn what each one does in a bit. Like I said, animal cells, so that includes humans, right? Dogs, elephants, giraffes, whatever, right? Cats, they all are made up of eukaryotic cells. Plant cells, also eukaryotic. Okay, so these are more advanced cell, more complicated, bigger, larger. The other kind, which is more primitive, more like simpler, those are the prokaryotic cells. They are smaller, they are simpler, they don't have a nucleus. So this is a bacterium, and you can see the DNA is just kind of in the middle, right? Okay, it's not contained in a ball, right? It's not contained in a nucleus. They have few types of organelles. Okay, so bacteria are prokaryotes. Okay, eukaryotes, prokaryotes. Complex, big, simple, small. Regardless if you're a prokaryote or eukaryotic cell, right, there is going to be a cell membrane, one of the common features, right, of all cells. You already know the plasma membrane is made up of phospholipids, right? You should know. Phospholipids. Okay, and it has a hydrophilic head and hydrophobic tail, right? So you actually have two layers of these phospholipids. Jamie asked if any of this new information will be on the test. Oh, you mean the quiz today? No, the quiz today is just lecture 1 and 2. Okay, this will be on like the next quiz though, right? Quiz number 2. Right. So the cell membrane function as a selectively permeable barrier. Right? Permeable means it will let things pass through. Selectively means not everything though. Right? You can control, you can regulate what passes through. Okay, and I already told you why that is important, right? So the phospholipid bilayer means two layers. You have one layer here, you have a second layer here. Basically, you have the hydrophobic tails, the hydrophobic portion that is sandwiched between the hydrophilic heads, right? So if you think about a cell, this is a cell. The outside of the cell is mostly water. The inside of the cell, the cytoplasm, is also mostly water. So they are hydrophilic environment, right? Which makes sense to have the hydrophilic head facing either outside or inside. If you look at the electron microscope picture here, that's what it looks like. That's an actual cell membrane. The two thick black lines, those are the hydrophilic heads. And then the white gap in the middle, that is the hydrophobic tails. Okay. So cell membrane is not something that's only found on the outside of the cell. You will also find cell membrane surrounding some organelles. Okay? Some organelles actually have two layers of cell membrane. All right, now this next couple of slides talks about various structure of the cell. I think it's best to learn this if we draw it together. Okay, so if you can get a piece of paper and then draw along with me, it would help you understand what goes where and the relationship between them. Okay, so get some paper, highlighters, colors if you have them. Okay, we're gonna draw a few things here, and then we can go back to the lecture notes. Okay, it's a little bit more interactive this way as well, right? So you're not just like watching me do it, right? Yeah, that's okay, no problem. Yeah, it's okay. That you can just watch the recordings, right? If you missed the beginning part. All right, so this is eukaryotic cells. All right, so here we go. Let's just start. Okay, so how big is this thing, right? Now this is your paper. Okay, we're gonna start here, and then we're gonna go all over the page like that. Okay. So I don't want you to do a tiny diagram in the corner of the paper. Okay. Use the whole paper. Use the whole paper. Right. So on the top left of your paper, I want you to draw a ball. There. Okay. So this is the nucleus. Nucleus. Okay. Now don't do the next part here. This is the whole cell, right? With the cell membrane, and then I'm just looking at the nucleus, right? But I'm gonna take away the cell membrane here, so we have more space. So nucleus is a feature of eukaryotic cells. You will not find a nucleus in a prokaryote, right? In a primitive cell like bacteria. Okay, here's the nucleus. Now, the nucleus is actually surrounded by a membrane. Okay, but the membrane is not going all the way around. Okay, the membrane has a little bit of gaps in between. Okay, so like that. I'm just gonna put some gaps there. Okay. Now let me show you another view here. Let's say this is the whole thing, and then I have the whole membrane like this, right? Surrounding it, the entire thing. Right? You would have some pores, some openings. Okay, and that's what these gaps are for. Those gaps are called the nuclear pores. Nuclear pore. All right. They have same shape. What have same shape?
Membrane.
The membrane has the same shape. What do you mean?
The same light, the same issue.
Oh no! Oh, I'm just trying to show that there are gaps in the
Okay.
membrane. Yeah. Think of it like this. Okay. Think of an apple. Okay. Actually, apple is a bad idea. Think of an avocado. Okay. You guys know avocado? You know avocado, right? So that's the avocado, right? The skin is like the membrane. Okay. But what's different here is now take a fork and then you poke some holes on all over the avocado. The holes are the pores. Okay? I don't know if that helps. So the nuclear pores, they allow things to enter or leave the nucleus. Okay? I'll show you the professional drawing later. You'll see it better. But I just want you to draw it to help you understand. Now this here is the nuclear envelope, okay, which surrounds the nucleus. Okay, but it has some gaps in it, it has some pores in it to help regulate what goes in and out of the nucleus. Okay, I hope you're drawing along. All right, it doesn't have to be pretty, but the process of constructing the cell from scratch, I believe, is going to help you remember it better. Now on the inside, we have an other structure, just like the avocado. There is a seed in the middle, right? So there is kind of like a seed in the middle of the nucleus as well. That is called the nucleolus. Nucleolus. Okay. Nucleolus. A circular structure that's inside the nucleus, and its job is to make an other organelle called ribosome. Don't worry about what they are yet. Just know that they are made in the nucleolus. Okay, there's going to be a lot of new terms in this lesson, especially if this is the first time you're learning the biology. So are we together so far about the nucleus? Right? Just think of the avocado. The membrane is like the skin. Poke holes in the skin, then you get the pores. The seed in the avocado is like the nucleolus. Okay? Fantastic. So what's in all this area, right? Okay, well, that is where you will find genetic material. Okay? So those are chromatin. Chromatin. Now I can hear you screaming at the screen. Hold on a minute. Isn't genetic material DNA? Right? Like what is chromatin? Okay, and then you must have heard of chromosome before, right? Like, so what is it? Is it DNA? Is it chromatin? Is it chromosome? Allow me to explain. Okay, so chromatin is basically DNA wrapped around proteins. Okay, medicine is correct. Okay, the DNA on its own is very long. It's unwound. Okay, and if you try to pack all that, if you try to shove all that into the nucleus, it won't fit, and it's gonna get tangled up. And when things get tangled up, they tend to, you know, break easily, right? They're not gonna be able to move very easily. So in order to fit them all into the nucleus, you have to take DNA and you have to package them. You have to wrap them around proteins. Okay. Think of a sewing thread, right? Okay. You don't just take a bunch of sewing threads and then shove it into the container, right? You have to wrap them around spools, right? Like this, right? You understand? Right? So now they are nicely packed together and you can fit a lot of them in an organized way. The red thread is not gonna get tangled up with the green one, and so on and so forth, right? Okay, and that's important because we don't want DNA from chromosome 1 to get tangled up with chromosome 7 or something like that. Okay, so DNA very loose, unwound. You want to package it a little bit with some protein, that becomes chromatin. However, when the cell divides, you need to make copies of these DNA, right, to pass on. That's one of the part of the cell theory, and you need to move them around. So you need to take the chromatin and condense it even more, and that becomes chromosome. Okay, so you only make the chromosome when you need to move them around. Okay, same as you, right? Like if you need to move from one place to another, you gotta pack your stuff, right? Okay, but once you move, you can unload them, you can take them out. Okay, so when a cell is not moving the genetic material, it's in the form of chromatin. Okay, and then when you need to move them, you pack it, condense it, it becomes chromosome. You move them to where you need them to be, and then it unpacks again in chromatin. Is that okay? So from level of compactness, right? DNA, chromatin, chromosome. So you will find it in the nucleus. All right. Now moving on over here, the nuclear envelope that we draw actually does not stop here. Okay, it will actually extend into the cytoplasm to form these long tunnels. Okay, long tunnels. Okay, and then it will continue. I'll use a different color here. Keeps on going outward into the cytoplasm. All right. So these networks of membranes, essentially, we call them endoplasmic reticulum. Okay. On your next quiz, quiz number 2, I will be showing you pictures of these organelles, and you would have to tell me what they are. All right, so make sure you know how to spell them. Okay, endoplasmic reticulum. Okay. Endoplasmic means inside the cell. Reticulum means a network. Okay, in this case, a network of membranes, right? Okay, so let's backtrack a little bit. Let me choose a different color here. In the nucleolus, okay, the seed, right, of the avocado, in the nucleolus, that is where you will make something called ribosome. The ribosomes, once you make them, will exit the nucleus through the pores, right, through the nuclear pores. So they will come out. The ribosomes will come out. Some ribosomes will be floating freely on their own. Others will attach onto the endoplasmic reticulum. Okay? So these things right here, they are just free floating. Free floating ribosome. Okay? Sloshing around in the cytoplasm. Okay, like a jellyfish, you know, just moving back and forth in the wave. Some will be attached onto this endoplasmic reticulum. When you look at this under the microscope, okay, electron microscope, right? The presence of the ribosomes on the ER makes it look rough. So if the endoplasmic reticulum has ribosome on it, we call them rough endoplasmic reticulum. Okay, with ribosome on them. If they don't have the ribosomes attached to it, then it's smooth. It has a smooth appearance, so we call it smooth ER. That's everything I want to draw for now. Any questions? Please ask. If like any of this doesn't make sense. All right, I will
Sir,
take. Yep, go ahead, please.
this is not going to be a part of this test today, right?
No, it's not. Yeah,
Oh, for the next one. Okay.
yeah, for next one. Mhm.
So this is lesson 3.
This is lecture 3, yes, which will be on quiz number 2.
Okay, thank you.
Okay. I hope you guys are really with me. All right. So this is the professional drawing. Right? You can see the nucleus here. There is the thing in the middle, the nucleolus. And if you look closely, you can see the chromatin here, right? The chromatin, right? Which is just DNA wrapped around proteins. You have the nuclear envelope, and then on the envelope, you can see all these pores, right? Nuclear pores. See, it should be much easier to pick out the different parts now that you have drawn it yourself, right? Okay. Hopefully you've drawn it. Okay. I don't know. It's hard for me to tell. And then the envelope is going to extend outward into the cytoplasm. Some of them will have some yellow balls on them. Yellow dots, right? What are the yellow dots? What do they represent?
Ribosome.
Ribosomes, that's right. That's the ribosome, right? And they came from the nucleolus, right? Okay, so this makes them rough ER. And then you have some ER that has no ribosomes on them, so they are smooth ER. All right. I have not told you what they do yet. Okay, that's coming. I just want you to know what the structures are and where they are and how they're related. Okay. So this picture shows you the packaging that I was talking about, right? So DNA, the double helix is very loose. You can wrap them around proteins. These proteins are actually called histones. I guess you don't really have to know it. And that becomes chromatin. If you package it further, that's when you have, that's when you have the chromosome. Okay, when do you get the chromosome? You only need the chromosome when you are dividing the cells, when you need to move them around. Okay. That just tells you more of the same thing. We have the nucleolus in the middle. That's where it makes the ribosomes. Okay, nothing new. So let's take a closer look at the ribosomes here. Okay, the ribosomes. It actually, it's really small, first of all. Okay, if you look at a cell diagram, right, like they are just these tiny dots compared to the nucleus, right? So like if the nucleus is like a basketball, right, then these ribosomes are just like a sesame seed, I would say. Okay, basketball and then sesame seed, very, very tiny, right? But if you zoom in, if you take a look at the structure of a ribosome, you will see that it has a large subunit and a small subunit. Think of it like a burger bun. You got the top bun and the small bun. Top bun is bigger, right? So that's what makes a ribosome. Usually they are apart. The large subunit and small subunit, they're usually apart. But when the ribosome does its job, they will come together. And the function of the ribosome is to make protein synthesis. Okay. Let's think back to lecture 2. You should know because you have a quiz today, proteins are made of amino acids, right? And you should already know you need to create a peptide bond to link the amino acid together, right? So what actually does that in the body? The ribosome. Okay, so the ribosome will decode information from your genes and it will know, okay, I gotta take this amino acid, I need to connect to this one, form the peptide bond, take the next amino acid, attach it until you get the whole protein. That's the job of ribosome. It makes protein. Okay. What is the ribosome itself made up of? Interestingly, the function is to make protein, but the ribosome itself is also made up of proteins. Okay, proteins and a little bit of RNA. Okay, remember there are 3 kinds of RNA, right? Messenger RNA, like the ones you know in the vaccine. There's rRNA, okay, which is the kind that you find in ribosome. In this picture, the lines here, the black lines, that's the endoplasmic reticulum. And all these dots that are attached to it, that makes it the rough ER. So that's the ribosome. Okay. So they are made in the I'm just gonna read it to you. It's basically the same thing I've been telling you for the past 15 minutes. Ribosomes are made in the nucleolus and exit into the cytoplasm through the nuclear pores. Okay. Some ribosomes exist freely in the cytoplasm, the one that are just floating around. Some are attached to the endoplasmic reticulum, right? Which makes it rough ER. Okay. This next slide is important. It will lay the foundation of what we'll be learning for the next couple weeks. Okay, all the way up to lecture 8, actually. This is called the central dogma of biology. Okay, dogma is like central theme, I guess you can say. Yeah, but I don't know. People like to use big words, weird words. Okay, it's not my word. Okay, that's actually what they call it. Okay, the central dogma, central theme of biology. All right. So to make proteins, we need to find the instructions, right? And where is that instruction located? How do you know how to make protein? What did I tell you before, huh? How do you know what amino acid to put together? Which part of the body tells you that? Have that information. Yeah, Madison said the DNA instruction, exactly, right? Okay, so you have the instruction to make the protein in the DNA. Okay, but DNA, as I told you, is really big. It's in the nucleus. It probably will not fit through the nuclear pores, right? And the ribosomes are all the way in the cytoplasm, right? So like it doesn't, you cannot take the DNA out of the nucleus. Okay, it's too big. And even if you could take it out, you don't want to, because like it might break down out of the nucleus, right? So what do? You make a copy of the DNA, not the whole thing, okay, but only the section that you need to make a particular protein. You make a copy of that in the form of messenger RNA. Okay, so this process is called transcription. Okay, so DNA, if you make a copy of it in the form of RNA, that's called transcription. Okay, think of it this way, okay? There is this textbook at the library. It has a lot of good practice multiple choice questions that you could use for studying your test, okay? But they won't let you sign it out of the library, okay? They say, oh, we only have one copy. We don't want it to get damaged. We don't want to get lost, okay? So what do? You photocopy it, right? You can photocopy it. Are you going to photocopy the whole book, right? All like, 800 pages? No, right? Because your test is not on all 800 pages, right? Right. So you only photocopy the pages that you need, and then you take it out of the library, right? So same situation here. You don't need to copy the entire 46 chromosomes because you don't need all the proteins at this very moment, right? So you only copy the section that you need, and you will copy it in the form of RNA. Okay, so the RNA is going to exit into the cytoplasm through the nuclear pores. And in the cytoplasm, the RNA is going to get decoded by the ribosome. Okay, so here you can see the ribosome, the large subunit, the small subunit. It's going to clamp onto the messenger RNA. It's going to decode the information, and then it will use it to build the protein. Each of these purple circle represent an amino acid, right? So this process is called translation. Translation. Okay, it's called transcription for DNA and RNA. Transcript means to copy, right? Because DNA and RNA are the same language. They are both nucleotides, right? And then from RNA to protein is translation, because it's a different language now. RNA is nucleotide and proteins are, hopefully you're thinking, amino acid, right? So that's why transcription, translation. Any question? Okay. Which of the following is a prokaryotic cell? Or prokaryotes, okay? Same thing. Okay. Thank you for those who participated. The correct answer is E. coli. That's the bacteria, right? Bacteria. E. coli, right? And bacteria are prokaryotes. Liver cells. Liver is found in us, right? So our cells are eukaryotic. HeLa cell, that was taken from the ovarian cancer. That makes it eukaryote. Amoeba, it's a unicellular organism, also prokaryote, right? Sorry, also eukaryote. So this is the bacteria. Do you guys know any other bacteria? You must have heard of E. coli, right? Anything else that you've heard of? Usually bacteria has a alphabet followed by something else. COVID is a virus, right? Mononucleosis is a condition, not quite a bacteria, but good try. Staph, that's right. Staphylococcus.
Malaria.
What's that? Sorry.
Malaria.
Good try. Malaria is a parasite. Oh, okay. I have a good one now from N. Okay. H. pylori. Have you guys heard of H. pylori? No, is estimated like, sorry, what's that?
No.
No, okay. It's estimated that over 35% of the population has H. pylori in their stomach. Okay, and it could give you stomach ulcer when they flare up. Anyway, I teach you about that next semester. Yes, from Augie. I'm sorry if I'm pronouncing your name wrong. Salmonella, very good. Okay, so like all these bacteria, right? They are prokaryotes. That's the point. Okay, what is the function of the nucleolus? What is the function of the nucleolus? I've been like telling you this repeatedly for the last 10 minutes. Please choose the right answer.
I can't see the choices.
Oh, it's blocking it again. Sorry.
Yeah.
Sorry. Okay, a little bit, sluggish this morning. You guys like not much participation. Maybe you guys up studying all night or something last night. Anyway, that's yeah, most of you got it right. Okay, it's the production of ribosome, right? That's where those little dots, those little hamburgers came from the nucleolus. All right, let me just think about how far we should go. Okay, we're just gonna go maybe another 10 slide or so, and then I will let you go. We are gonna learn about the endomembrane system. Endomembrane. Endo means inside. All right. So in this case, inside the cell. We're gonna look at a network of membrane that exists inside the cell. There are a couple of components in the endomembrane system. I'll show you what they are. You already learned some of them, and they have interrelated functions. Okay, that allows the cell to do various things, including synthesis, which is production, processing, and digestions of things like lipids, proteins, and other large molecules. Okay. The endomembrane system is also responsible for recycling and disposing things that we don't need, okay, or things that are harmful in the cell. That's what it is. The key components of the endomembrane system are listed here. There's the nuclear envelope, which we drew, and the nuclear envelope extends into the cytoplasm, right, to form the rough and the smooth ER, depending on whether they have ribosomes on it. And then there's something called Golgi body that we haven't talked about yet, and then there's something called lysosome that we haven't talked about. Okay, so let's start with the rough ER. All right, so that's this part right here, right, with the yellow dots on them. That is continuous with the nuclear envelope. It's attached to it. Okay, it's just an extension of it, right? And it's covered with ribosomes. Nothing new so far. Same thing I've been telling you over and over again. But what does it do? The ribosomes will actually produce the protein, synthesize the protein directly into the inside of the rough ER. And then it's going to get packaged into a vesicle, which will then pinch off from the rough ER. Now that all sounds very abstract, right? Let me try to explain to you here. Okay, you don't have to draw this. You can just watch. Where is it? So like this is like the edge of the rough ER. Say that again. Right, that's a rough ER right here. Now this is the inside and this is the membrane that surrounds the ER, right? We're gonna have our ribosome right here. That's the ribosome. Okay. I'll post this afterwards, right? There is gonna be the messenger RNA that it's decoding, right? And then you will make the protein, right? You make the protein. So the protein will go directly into the inside of the ER. Once you the ribosome finish making the protein will fold up into the 3D shape, right? You know, the secondary, tertiary structure. It will fold up. And then it will move towards the edge of the ER. And then this is what's going to happen. Okay, I'm going to draw another picture. What happens is, like this part here, it's going to start to squeeze a little bit. Okay, with the protein inside. Okay, I'm just trying to show you like in a series of diagrams of what's happening. And then it's going to pinch off. Like this. So that is a vesicle. Can you visualize this? Like I usually don't show videos in the lecture because then when I post it onto YouTube, somehow Google is able to detect there is like a video that belongs somewhere else in the internet, and they say it's like copyright violation, and they won't let me post a lecture, right? That's why like I cannot show you an animation. But go to the video resources, right? You will see some animations there. Is this okay? Yeah. So like think of like Play-Doh, right? Like you take a ball and then you pull it off. Okay, that's basically what it does. So the rough ER and the ribosome works together to make proteins and package them into a vesicle. Okay. Now, where is this vesicle going to go? Let me just take a look at my slides and see if okay, so I'm gonna skip the smooth ER for now, and I will tell you where the vesicle will go. All right. So let's go back to here. Right, now you can draw along if you want to. Let's add a new organelle here. Right, there's a new organelle, like maybe over here. Right, and it looks like this. It's like a bunch of pancakes together, like that. Yeah, so you can draw that first. Just give me a moment. I need to make more space for myself. Gotta put it over there. I don't. Okay. So this here is called the Golgi body. Golgi body, or sometimes called Golgi apparatus. Doesn't really matter. Yeah, you can call it both. All right, so it's like layers and layers of membranes. All right. So if you were with me the whole time, there is going to be a vesicle that pinches off, right? That butts off from the ER, right? And that vesicle is going to have some protein in it. Right? So this is vesicle with protein. Yeah, you could draw it if you want, okay? Kiana? You should, yeah, you should draw it. I'll give you guys a moment. Sorry. Okay, don't worry. I will end at 10, okay? For those of you who are wondering, when is this gonna be over? Okay, so this vesicle with the protein in it is going to go to the Golgi body. You see the protein that we just made is not quite ready yet. Okay, sure, it folds up into a 3D shapes, but then you still gotta make some modifications to it. Okay, I'm not gonna go into why, but you know, like it's not quite a final product. It's like after you bake a cake, right? Like you can't just serve that. You gotta, you know, add icing to it, you know, put some fruits or whatever, right? You gotta make it look nice. You gotta make it like the final product, right? So that's what the Golgi body does. The Golgi body is going to receive the vesicle, and then, right, it's going to sort, modify, and repackage the protein. Okay, think of it like a post office. You got all these mails coming in, you got to sort them, right? Okay, you got to patch them up if there's something wrong with the parcel, and then you might have to repackage it and then send it out. That's what the Golgi body does. Okay, they sort, modify, and repackage vesicles. Repackage the contents, we will say the contents of a vesicle. Okay, so essentially it's the protein, right? Yeah, like a packing plan of a cell, if you would. Okay, distribution center. Okay, so this vesicle is gonna go to the first layer, and then you know it will get passed on to the next layer and next layer, and sometimes they go back and forth a couple times, and then eventually you will have a brand new vesicle. What color should I use? Let's use green. Brand new vesicle. And then the protein is going to be like modified now. Okay? Any question? All right. Now this next part is important. Okay. Where will this go? What are the destinations of this thing? Well, there are several destinations. Okay, there are several destinations. I forgot how many, like 4 or 5. Anyway, it doesn't matter. We'll write them as I remember them. Okay, so first of all, this protein may be ready for use immediately. Okay, so it will be released from the cell. Okay, maybe it's insulin, right? Maybe you just had a meal and you need to release the insulin into the bloodstream. So this package with the insulin inside, it will go to the cell membrane, and then it will let it out of the cell and use it right away. Okay, maybe you don't need it. Maybe you don't need it. So it will stay in the cytoplasm until it is needed, right? Sometimes we cannot make things on demand in the body. Okay, like it's like too rushed, you just can't get it quick enough, right? So like you want to have some of them stored, and then release them as soon as you need them. Okay, now this next part, it might not make much sense right now, but it will after lecture 4. Sometimes this will become part of the cell membrane. Okay, it will become part of the cell membrane. Right? So we know the membrane is made of phospholipid. We know it has a little bit of cholesterol, but it turns out it also have a bit of protein as well. Okay. And one last possibility, it can also become something else called a lysosome, right? Which I will explain in a bit. Okay. So just to quickly recap, the rough ER, which has the ribosomes on them, will work together with the ribosome to make protein, package them into a vesicle, which you pull off from it. It pinches off, it butts off from the rough ER. But this is not the final product. Guess it has to be modified, right? So it goes to the Golgi body, it gets passed, back and forth a few times. You sort them, the Golgi body sorts it, modifies it, repackages it, and then you have a new vesicle, right, with the modified content on the inside. What's going to happen to this vesicle? You can release it from the cell right away if you need it. You can store it for later use, right? It could go to the cell membrane and become part of the cell membrane, or it could become another kind of organelles called lysosome. Do we have any question? When I'm in person teaching, the same thing. Yesterday I asked any question. Everybody just smile and then don't say anything. I don't know if you guys are actually with me here. Nia said, will there be a diagram of prokaryotic cell? No, we're just doing eukaryotic cell. Okay. All right.
Will we have any assignments on this labeling these?
There are no assignments, guys. A lot of you are emailing me and asking me, is there an assignment? There's no assignment, okay? There are only two things you have to hand in the course, lab 1 and lab 2, okay? And you will know. Yeah, look at the schedule and you will know when is it due. But then I will be telling you as well, right? When they're happening. Like I'm gonna have to tell you what to do for the lab as well, right? So don't worry about them right now. You just have to worry about a quiz. All
okay, right? So we don't have any lab.
Nothing due. Yeah, nothing due.
Okay,
nothing
cool.
due. Yeah. Okay. Well, let's go back to our slides here. Okay. So we have the rough ER. This is the professional drawing of what's going on, right? It pinches off. Where is this thing gonna go next? Now, hopefully it's gonna go to the Golgi, right? The Golgi body. This is about the smooth ER, which we'll skip for now. I'll come back to it probably next class. This is the Golgi body that I was telling you. The Golgi body is layers of membrane stacked together, like pancakes, one on top of the other. It receives vesicles produced by the rough ER, like the one that butts off, right? And then it's going to get sorted, repackaged, tagged, right? Tag means it tells it where to go, right? Where will the protein go from the Golgi body? I already wrote down for you, okay? The different paths. Let's take a look at the lysosome now, okay? Because that's one of the possible destination, right? Okay, the newly packaged thing, it could become a lysosome. So lysosome is also part of the endomembrane system. And what it does is that you can think of it as like the recycling center, okay? Like the garbage collection facility, okay? The lysosome contains digestive enzymes. We know what enzymes are now, right? They break down macromolecules, right? But they can also help recycle old organelles. And so let's say you have an organelle that's not quite working anymore. The lysosome will come and fuse with it, and then the enzyme inside will break down the worn out organelle. Okay, so that way there's no malfunctioning, right? And you can unlock the nutrients that is, trapped in those organelles. Sometimes, okay, some cells will eat things from the surrounding. Your white blood cells, they do this, okay? They could eat up a bacterium or something like that. And then you want to break down the bacteria, right? So here, the food, right? You think of it as like bacteria. It goes into the cell and you need to break it down. So the lysosome will fuse with it and digest it. Okay, so they break things down. That's the first function. Second function, the lysosome can, in some situation, releases all of its enzyme in the cell, and that will cause the cell to die. Okay, this is called apoptosis. Program cell death. Okay, program cell death. In some situations, it is necessary to kill off cells deliberately in the body. Okay, for example, if a cell is too old and then it's like getting a little bit wonky, it's doing things it's not supposed to do, you want to kill that cell off, right? Okay, so the lysosome will release its content, trigger apoptosis, and destroy that cell. During embryonic development, did you know at some point when we're still inside the womb, we have webs between our fingers? Okay, we have webbed hands. All right, so now obviously we don't, which is a good thing. Otherwise, it's like a lot difficult to, grab things and stuff like that. So we want the cells between the fingers to die off, right? Okay, and that's when the lysosome will be useful. It triggers apoptosis in those cells. So the web, the cells in the web, they die off, the web is gone, and then we have free moving fingers. Okay, this is day 51 in the embryo. Can you believe it? Okay, last slide, and then I will let you go. Because the lysosome contains a mixture of enzymes, right? And enzymes are proteins, and proteins, the instruction to make them are encoded in the DNA. So it's possible for people to have mutations in the DNA that cause them to be missing some enzymes in the lysosome. Okay, there's a lot of different enzymes in it, and if the instruction to make one of them is mutated, then you'll be missing that enzyme, right? So it turns out there is a group of conditions called lysosomal storage disease, and depending on which enzyme you're missing, the outcome is different. I'm just going to show you two. Okay, there's first one is called Pompe disease. This is when you are missing an enzyme in the lysosome that breaks down glycogen. Okay, and then they start to build up to a harmful level in the muscle cells. Okay, and this is going to be dangerous, affecting the heart muscle, right? The heart is going to be affected, right? So that's one example. If the heart muscle cannot beat properly, right, then you have a heart condition, right? Basically, that's what I'm trying to say. Second one is called Tay-Sachs disease. You might have heard of this. This is one of the things that they screen for in prenatal screening. It is a situation where the enzyme that is responsible for breaking down lipids is missing in the lysosome. And that causes the lipids to build up excessively in the brain cells, which interfere with brain development. Okay, so there are a whole list of them. If you search up lysosomal storage disease, and Wikipedia will show you like a whole list, like 28 of them or something like that. All right. Well, look at that right at 10 o'clock. Right. I still have to do the smooth ER. That's okay. I'll do it next time. But hopefully this gives you a good overview of what's going on. Now, next class, we will finish lecture 3. And we probably will start number 4. Okay, we will have time for that. So please print the notes accordingly. And good luck with the quiz today. Okay, I look forward to grading it. Okay, to see how well you guys do.
Okay, sir.
Yeah.
For the quiz using the lockdown browser, we don't have to take a picture of the answer. Yeah, we do, don't we?
You don't have to take a picture of anything. You will just be doing the test directly in your browser.
Okay, and when it's finished, we can submit it there and don't have to submit it anywhere else.
That's right. Now, Madison is saying that's for math, so maybe you want to chat with your classmate to Yeah, but for me, you don't have to take picture for anything, okay?
Okay,
But
okay,
maybe
that's
you
not a
do have
problem.
to do it for your math class, okay?
Yes, I know we have to do it for math, so I was wondering
Yeah,
if we
for
have to do it
math,
for
it's the same thing. Yeah, you have to take the pictures afterwards and then